Yes, audible. So transcription first is completed last class, isn't it? Jyoti Kalyan. My voice audible or not?
Yes, sir, audible.
Right. In last class we completed transcription. Transcription in prokaryote, transcription in eukaryote. All that told you just now the transcription. What is transcription?
Synthesis of DNA. Sorry,
Right.
from DNA.
Synthesis of RNA from the DNA strand. Next followed translation. In translation on the prepared RNA strand, preparation of polypeptide chain of amino acids. This polypeptide chain of amino acids called as protein. So the total process called as protein synthesis. In part of in protein synthesis, one stage is transcription that was completed, and second stage is translation. Let's see today translation process. In our book given only prokaryote translation, only prokaryote. But anyway, I will include some important points of eukaryote translation also. Right? Let's start transcription process. Sorry, translation process. Just 1 minute. Sorry for inconvenience. Am I audible? Hello?
Yes, sir.
Right, just wait. Prokaryote. Are you able to see this slide? Hello?
Yes, sir.
Right. Prokaryotic translation, right. Actually, in our book given only 4 stages of translation in eukaryotes. What are the 4 stages? Do you have book?
Yes, sir.
What are the
Sir,
four
which
stages?
chapter? It is which chapter, sir?
Prokaryote translation in protein synthesis.
Okay,
In
okay.
protein synthesis, I think unit number in third
9, sir.
block, right. Third block, unit number 9. Did you turn on the page?
Yes, sir.
Right. Okay, actually in our book, given only initiation, elongation, peptide bond formation, termination, right?
Yes, sir.
So initiation, here also same, the stages, initiation, elongation of polypeptide chain, peptide bond formation, and termination. But initiation sub-stages also there, but which are not given in our book. Just
Yes, sir.
write down in initiation in clearly. Do you have pen or paper?
Yes, sir. Yes, sir.
Right. So first of all, in translation, definition completed. Translation mean preparation of polypeptide chain of amino acid on RNA, mRNA stand. The process called as translation. In translation process, prokaryote involved 4 stages such as initiation, elongation, peptide bond formation, and termination. But in initiation, the again two subdivisions. Are you able to see this slide?
Yes, sir.
Right. Initiation, initiation of in initiation first we should activated amino acid. Later, this activated amino acid transferred to the tRNA. Before that, before that, let me tell you in what are the requirement for translation. Translation definition clear. Definition preparation of polypeptide chain of amino acid. What are requirement for preparation of amino acids chain? What are requirement? What are requirement from translation? Listen, translation requirement. First of all, tRNA, transfer RNA, mRNA stand, already mRNA stand prepared, right? Where are the tRNA? First list out the names. First of all, requirement from translation. Here mRNA stand, need, hello, follow? Are you follow? Hello?
Yes, sir. Yes, sir.
Right. The requirement first mRNA stand, write down. Next tRNA, next ribosomes. These are the major and amino acids also. mRNA, tRNA, ribosomes, amino acids, and translation enzymes. I will explain later what are the translation enzymes. Translation enzymes, right. So first of all, how we get mRNA? How we get mRNA? Hello?
Yes, sir.
How we get mRNA?
Uh,
By which process?
Protein synthesis? No, no. I- Initiation, I think.
No. Transcription. Transcription. Already told you protein synthesis, two stages, transcription, translation. Transcription from the DNA, preparation RNA. Translation and RNA stand prepared protein. That is translation. One, now we are discussing translation. Before entering into the translation, we checked what are the requirement for the translation process. Already told the requirement are RNA. My question, how RNA available? RNA prepared. The RNA prepared from the DNA strand through the transcription process. Next, another requirement is tRNA. Where are the tRNA? tRNA present in the cytoplasm. Where are amino acids? Amino acids also present in cytoplasm. Ribosomes also present in cytoplasm. They are all present in cell cytoplasm. Right? So what is the function of tRNA? Transfer RNA. Name itself have transfer. What is transfer RNA? tRNA. Which one transfer? Hello?
It transfers the amino acids.
That's it. Transfers amino acids. To transferring amino acids, it needs some energy. It needs some activation. It should be activated. So that is the first step. That is the first step of activation. So the activation amino acids, actually these amino acids are in inactivate state in the cytoplasm. They should be activated. Here are you follow the slide? Hello?
Yes.
Right. Right. Actually, this is inactivated amino acid. Follow my cursor. Inactivated amino acid. This inactivated amino acid with help of ATP energy. The activation is done by amino acyl synthetase enzyme. Here the enzyme involved, that is amino acyl synthetase. Write down amino acyl synthetase enzyme. Here the a high energy SL bond is formed between the phosphate of ATP and carboxylic group of amino acid with the formation of amino acyloadenylate. Amino acyloadenylate, here adenylated bond. Here the first of all, this is inactivated amino acid. Inactivated amino acid, this is carboxylic group. Carboxylic group, this carboxylic group bond with ATP, TP, adenosine triphosphate. T for triphosphate. Here, how many phosphate? One. This is gamma phosphate. This is beta phosphate. This is alpha phosphate. Gamma, beta, alpha. Here the bond formation between alpha phosphate. Here, this alpha. Are you follow? Are you able to see this marker line, arrow mark, red line, arrow mark? Hello?
No, sir, it's not visible, sir.
Which
Initiation slide is there, but your cursor is not there.
in initiation, are you able to see the activation of amino acid, transfer of activated amino acid to specific tRNA?
Yes, sir.
Right?
Yes, sir.
Just do it. Pen visible?
Yes, sir. Now it's visible.
Right. So this is carboxyl group of amino acid. This is alpha phosphate group. So here the bond formation between this. As a result, one phosphate group remove. This phosphate group, alpha phosphate group remove. Now adenylated amino acid formed. The same amino acid carboxylic group, here the carboxylic group with phosphate. Now this phosphate group transfer to the amino acid. As a result, here the formation only 2-phosphate. That is called pyrophosphate. Diphosphate, ADP. Here the ATP converted into ADP because one phosphate group transfer to the amino acid. Now this amino acid activated. So that is the activation of amino acid. Which enzyme involved in this? To transfer a phosphate group to inactivated amino acid to bond formation, which the bond is high energy SL bond. The to formation SL bond with SL bond, which enzyme involved here? Which enzyme involved?
Yeah, amino acyl synthetase enzyme.
Right, amino SL synthetase enzyme formed. Now the amino acid is activated. Now second step, the activated amino acid. A means amino acid. A for A abbreviated amino acid. Two specific tRNA, right? So this is tRNA. This is tRNA. tRNA hydroxyl group 3 and OH group. 3 and OH group. Here 3 and OH group bond formation with carboxylic group of amino acid. Carboxylic group. So here the two bond formation. One is one phosphate also bond formation with carboxylic group, and tRNA also bond formation with carboxylic group with ester bond. With ester bond. Here the 3 end of tRNA high energy ester bond formation takes place. Hello.
Yes, sir.
As a result, amino acyl tRNA complex is formed. This is this is this is amino acyl tRNA. Amino acyl tRNA complex or charged RNA. This is charged rRNA. Charged rRNA means it amino acid carboxyl group. Amino acid carboxyl group bond formation with hydroxy OH group with ester bond. With ester bond. So the first bond is SL bond, right? Hello?
Yes, sir.
Hello? First bond between carboxyl group and alpha phosphate, here SL bond. The second, the activated tRNA, next bond with tRNA because the tRNA brings amino acid towards the mRNA strand to formation polypeptide. But here also the bond formation between tRNA 3-end hydroxy group to amino acid carboxyl group, the bond is ester bond. Right? Is it clear?
Yes, sir.
Now, initiation. Initiation of polypeptide chain. Initiation of polypeptide chain. In initiation of polypeptide chain required ribosomes. Here as it prokaryote, sorry, as it prokaryote, the 70s ribosome in, required. I already told you another requirement is ribosome. So two requirements are completed. One is amino acids, tRNA. Both the amino acids and tRNA present in cytoplasm. But the first of all, these amino acids inactivated state. When these inactivated amino acids they react with ATP energy activated. The activated amino acids bond with tRNA. Now tRNA complex form or activated tRNA form. Now another requirement is ribosome. To initiation polypeptide chain, ribosomes also required. you know already the prokaryote ribosome 70s ribosome, eukaryote ribosome 80s ribosome. We are discussing prokaryote translation. So the prokaryote ribosome 70s ribosome, the 70s ribosome two subunits. These are very important to learn translation process. Here the two subunits are small subunit 30s and large. Hello,
Yes,
slide
sir.
is
We are listening.
slide visible or not? Ribosome slide.
No, polypeptide chain is there, sir. Yes, it's
Initiation of
visible, sir.
polypeptide chain, prokaryotic ribosome 70s.
Yes, sir. It's visible.
Just wait. Right? Are you follow marker?
Yes, sir.
Red
Yes,
color.
sir.
Right. So
Yes.
this is actually 70 years. In 70 years, two subunit. 50 years, one is 50 large subunit. 50, this is large subunit. And 30 small subunit. Again, the 50 large subunit subdivided subunits are 5s rRNA. It made up of 5s rRNA, mainly ribosome made up of rRNA, that means ribosomal RNA and protein. So the 5s rRNA and 23s rRNA and 34 proteins. 34 proteins. And 30s RNA, 60s rRNA and 21 protein. 21 proteins. So this is about 70s RNA. Right? Next one. In initiation of polypeptide chain, before that first, again two stages, that initiation, 30s initiation complex, formation of 30s initiation complex, next formation of 70s initiation complex. First, let's see formation of 30s initiation complex. To initiation of polypeptide chain, here the required some factors. They are IF1, initiation factor 1, just wait. Initiation. Are you follow slide? 30s initiation complex.
Yes, sir.
Right now. Right. The required three initiation factors to initiation of polypeptide chain. Initiation factor 1, initiation factor 1 here, initiation factor 2, and initiation factor 3. Initiation factor 1, 2, and 3. They are required. Let's see the importance of this initiation factor. First of all, actually, the ribosomes, this is the ribosome complex, 70s ribosome complex. Before dissociation, initiation factor 3 attached to the E site. Here the ribosomes, a ribosome, three sites are there. This is very, very important. Just listen carefully. E site, P site, and A site. The first of all, initiation factor 3 attached at E site. As soon as the attachment of initiation factor 3, immediately the 70S ribosome disassociated as a 50S ribosome, 50S subunit and 30S subunit. Clear? First step clear? Hello?
Yes, sir. Yes, sir.
Right.
Yeah. What is EPA, sir?
Peptidyl site. This is peptidyl, P for peptidyl site, A for amino
Okay.
acyl site, E for elongation site. So we will
Okay.
discuss later each and every one. Okay? Right.
Okay. Okay, sir.
So let after dissociation into 30S and 50S, later in second initiation factor 1 attached at A site, attached at A site with help of GTP energy, with help of GTP guanosine triphosphate energy, IF1 attached at A site. Right? Next,
Yes, sir.
third step, initiation factor 2. Initiation factor 2, actually this initiation factor 1 block the A site. Initiation factor 3 block the E site. Now which is remain empty?
P side.
Only P site remain empty. Now initiation factor 2, with help of tRNA, tRNA, this is amino acid, this is amino acid, this blue color is tRNA.
Mm.
Already we discussed earlier slide how the bond formation between the activated amino acid to tRNA. Already we discussed in previous slide.
Yes, sir.
Activated, how activated amino acid with help of ATP, that is also completed. This activated amino acid attached to the tRNA, that is also completed. Here attached to tRNA at the 3 end of hydroxy group with which bond? Bond name ester bond, with ester bond.
Yes.
So this
Yes, sir.
tRNA, now this tRNA complex brings toward the P site with help of initiation factor 2. Initiation factor 2 carries this amino acid tRNA toward the P site. So, always remember this first amino acid methionine. First amino acid methionine. This is N-formyl methionine. First amino acid. N-formyl acid. Now now this is called 30S initiation complex. This is called 30S initiation complex. Now clear? Hello?
Yes, sir.
Right. Later, initiation factor 3 removed. Here you see in last slide, is there initiation factor 3?
No, sir.
No, initiation factor 3 removed. So next means, what is the function of initiation factor 3? First of all, initiation factor 3 function disassociation. What happen if the initiation factor 3 absent in this complex?
Dissociation will not
Right.
occur.
That means there is a chance to association with large subunit, right?
Yes, sir.
Right. Or clear? Second slide. 70S initiation complex. Are you able to see?
Yes, sir.
70S initiation complex, right?
Yes, sir.
70S initiation complex. Same. Uh, What we completed last slide, as the initiation factor 3 option are removed, there is chance to added by larger subunit, right? Hello?
Yes, sir.
Right. Here the 50th larger subunit with help of mRNA stand. This is mRNA. This is this stand, mRNA stand. The this violet color. The mRNA with mRNA stand attached to the 30s RNA. Sorry, 30s subunit. Because here no restriction. What is the function of IF3? IF3 dissociation. If the absent of IF3, there is a chance to again reassociation. So as the absent of initiation factor 3, the larger subunit along with the mRNA strand bind to the 30s. Bind to the 30s. Actually, where exactly it bind, here the binding with actually this mRNA, the specific sequence are there to find out 30s mRNA. To find out for 30s mRNA, here the mRNA, sorry, 30s subunit, the mRNA have the specialized sequence. The specialized sequence called as Shine-Dalgarno sequence. Shine-Dalgarno sequence. So the Shine-Dalgarno sequence present in mRNA strand and complementary sequences present at rRNA. 30th ribosome, 30th ribosome in the part of 60th rRNA, 60th rRNA at 3 and Y-hydroxy group. Here the complementary. Here the sequence is AGG, AGG. This is the sequence. Shall-Dalgarno sequence. This is the specialized. Where these specialized sequence are there, there exactly mRNA stand bind, where exactly 30s subunits bind, right? So now for example, this is mRNA stand. This is Shile Dalgarno place. This is actually starting site. That means initiation site. Polypeptide chain initiation site. Before initiation, there is specialized sequence are there. The specialized sequence called as Shile Dalgarno. This Shale Dargarno 30s subunit exactly bond. This 30s sequence also in the 60s rRNA, this sequence UCCU. Because here mRNA A, G, G, A, where this repeated sequence present, that is considered as a Shine Dalgarno. Shine Dalgarno is scientist name who discovered this specialized sequence. That's why the name given as a Shine Dalgarno sequence, right? So anyway, 30s ribosome and mRNA and also closed with larger subunit also, 50s subunit also, 50s subunit also, right? Is it clear? Hello?
Yes, sir.
Right. So now this 30s subunits, now later one by one removed. Already initiation factor 3 removed. Next, initiation factor 1 and initiation factor 2 removed. In this first, initiation factor 1 removed, later initiation factor 2 removed. If you see here in third figure, there is no initiation factors. Already we can observe here the first initiation factor 1 here, initiation factor 1 removed, and here initiation factor 2 also removed, right? Now without any initiation factor, this is considered as a 70s initiation complex. What are present in 70s initiation complex? In 70s initiation complex, 30s subunit and 50s subunit with the tRNA amino acid. With the tRNA amino acid. No other initiation factor. So anyway, initiation complex, both initiation complex started. Well, first of all, 30s initiation complex Second one, 70S initiation complex. And the initiation site also recognized. Initiation site also recognized the initiation site is Saint Dalgarno. The Saint Dalgarno place is upstream toward the starting site. For example, this is the starting site of the process. This side is the upstream. This side is the downstream. So Saint Dalgarno place is upstream. So upstream of the starting site. Is it clear?
Yes, sir.
Right. Right. Be careful. Each and every step very, very important. If you not understand any one stage, any one slide, you won't able to understand further. So be careful. If you have any doubt, ask me. Okay?
Okay, sir.
Right. Next one. If you see same initiation factors in eukaryotes, there are many different initiation factors are there. Their initiation factor EIF1, EIF2, EIF3, EIF4, EIF5, EIF6, and EIF4A, EIF4F. So these multiple are present, and but in prokaryote only three, IF1, 2, and 3. But the complex initiation factor present in eukaryotes. And another one is the another different is um, AGG complementary between mRNA and 60S ribosomal RNA. Already told you the Saint Dalgarno region is same. This Saint Dalgarno region is absent in eukaryotes. This Saint Dalgarno specialized sequence is present in prokaryote only. In eukaryotes, the small subunits of ribosome recognize the cap present at 5 end. Cap presented 5 end, only 5 end cap presented. The cap absent in prokaryote. As the cap absent, here it needs some specialized sequence to recognize the RNA. The specialized sequence is Saint Dalgarno sequence, right? And a tRNA charged with methionine binds to the AUG, which is located small ribosomal subunit. Next, initiation. So this is initiation process. The one difference is between the prokaryote and eukaryote. Prokaryote Saint Dalgarno specialized sequence present to recognition, but in eukaryote no need to recognition because here the specialized cap is present at 5 end in eukaryote. That's why no need any specialized sequence, right? And apart from that, here the initiation factors only three present in prokaryote. In eukaryote, more than three different type of initiation factors are present. That's it. Next one. Is it clear? First of all, initiation process clear.
Yes, sir.
Now elongation process. Now elongation. Okay, 70th initiation. Are you able to see the slide?
Yes, sir. Elongation.
Second slide, elongation. Right. Just see the first slide. Before that, in E site, P site, A site, which one empty? Are you able to see 70th initiation complex slide?
Yes, sir.
Right. Here, just wait. P site with tRNA, amino acid tRNA, right?
Yes, sir.
amino acyl tRNA. What about A site? Amino acyl site, this site, A site. Is it empty or filled with another tRNA?
No, sir, it is empty.
It is empty. Now elongation started. In elongation, slide clear? Elongation
Yes, sir.
complex. Right. Before slide, this A site is empty, right?
Yes, sir.
Hello? Right now, this A site, amino acyl site, amino site also filled with another tRNA, amino acyl tRNA, right? So before that, in elongation, prokaryote, here the some elongation factors are required. The elongation factors are elongation factors TU1, second one elongation factor TS, and third one elongation factor G. These three are required for the elongation. The first of all, elongation factors TU with help of GTP brings another appropriate amino acyl tRNA toward the A site. Already a P site with the first amino acid, methionine amino acid. First amino acid in the P site. Second amino acid, A site. Remember, always in translation process, first amino acid bind at P site. Rest of the all rest of the coming amino acids, all coming amino acids bind at A site and translocated into the P site. Remember this one. Clear? Hello?
Yes, sir.
The first amino acid always bind with in any translation process. First amino acid bond with P site. Second amino acids bind with not only second, the rest of the all amino acids bind at only A site, not P. Bind at A site and translocated into the P site. We will discuss later. So anyway, the second amino acid with help of elongation factor TU and guanine triphosphate GTP energy reached the A site. Later, here the peptide bond formation between first amino acid and second amino acid. The first amino acid and second amino acid between carboxylic group and amino group. First carboxyl group to second amino group. Let's see the bond formation. This is first codon. For example, here shine Dargonu sequence for suppose. Later, the move this ribosome move towards the where exactly starting codon present. Here is the starting codon, and this is first amino acid. First amino acid. This is second amino acid. Second amino acid. Now the bond formation between carboxyl, this is CCOO, C double bond carboxyl group here. Is it visible to you? Hello?
Yes, sir, it's visible.
Right. In the carboxyl group, here second amino acid amino group. This is amino group. So as a result, the carboxylic group and amino group. This is called peptide bond. Peptide, this is bond, peptide bond. The clear image in here is first amino acid. First amino acid, this is second amino acid. First amino acid carboxyl group. this OH and H, released in the form of water, released in the form of H2O, released in the form of water. Now C double bond O and NH, this bond formation. This is amino group, carboxylic and amino, both bond by this bond called as peptide between first amino acid and second amino acid. The enzyme name here involved peptidyl transferase. Enzyme name is peptidyl transferase. Is it clear first? Hello?
Yes, sir.
Right. Now you can imagine here the yellow color, this is the first amino acid. Here the bond formation with second amino acid. What happened later? What happened later? The first tRNA, the first amino acid tRNA translocated into E site here, this first tRNA. And A site, second tRNA translocated to P site, which is carried out by EFG, elongation factor G, with help of elongation factor G. Is it clear first? Hello?
Yes, sir.
Don't get any confusing here. First of all, P site filled with already first amino acid. In elongation process, when the EFTU, elongation factor TU, brings the second amino acid with help of GTP toward the A site, now elongation started. Later, the bond formation between the first amino acid to second amino acid, the bond name is peptide with help of peptidyl transferase. The bond formation between first amino acid carboxyl group, second amino acid amino group, by removal of water, hydroxy OH and hydrogen. For removal of water, one water molecule, here the peptide bond formed. Right, like this peptide bond formation continued until end. So this is, for example, first amino acid, second, and it comes the here the A site MTR filled with tRNA. Now in this slide, hello,
In the last slide, it is empty, sir.
empty. There is no tRNA. Now
Yes.
with help of EFTU, actually this EFTU activated with help of EFTS, both this one indirectly involved to activated EFTU. With help of EFTS, EFTU activated and bring amino acids toward the A site. Actually, this EFTU convert the GTP with help of the presence of EFTS. Right? Now the GTP converted into GDP and removal of EFTU. EFTU also separated. Now this EFTU activated with help of the EFTS brings another tRNA. Before that, EFG in elongation factor G translocated the A site tRNA toward the P site. With help of EFG, this A site here, what happened here? The A site tRNA present or not in first figure? Hello?
It is present, sir.
Present. But after influence of the EFG, it will be vacated, translocated toward the P site, as well as P site amino acids, P site tRNA also translocate to A site. First, immediately these two are in tRNA, one tRNA, second tRNA, with help of EFG, move left side, this side. That means first tRNA occupy the E site and second tRNA occupy the P site. As a
Yes,
result,
sir.
A site, right? As a result, A site will be empty. Now we can guess what happened next. What happened next? Hello?
Yes, sir.
Next, what happened? What can you assume? Just imagine. Already told you, already given a hint in just now. The only first amino tRNA bind at P site. Rest of the all coming amino acid tRNA bind at A site. Right?
Yes,
Hello?
sir.
Now, with help of initiation factor to TS, third tRNA amino acid, occupy the A site. After occupy the A site, what will happen?
It will translocate to P site.
When it is translocated, before translocated, what happened?
GTP converts into GDP.
And
With the EFG factor,
hmm, where is attachment to transfer to P site? Is
P
there
site,
any is there any connection?
mm,
Just see the first one, first two figures. These two figures. They're the first amino acid, right?
yes,
Second
sir.
amino acid, right?
Yes,
If
sir.
the third come, if the third one comes and occupy the A place, it should be bond formation between second amino acid to third amino acid, right?
Yes, yes.
After formation, which bond?
Peptide bond.
Peptide bond. After formation, peptide bond. Now, then translocated to P site.
Yes.
What happened next? Next, after translocation, after empty A site, what happened?
P will translocate into E.
Okay, next. Simple. The another tRNA amino acids occupy the A site. This is the. It will be continued until reach the nonsense codons. It will be continued until reach the nonsense codons. You don't get confused here. Just listen. For first figure, how you follow first figure?
Yes, sir.
In first figure, P site amino acid, right?
Yes.
A site also with amino acid, right?
Yes, sir.
This A site amino acid occupied with help of the EFTU GTP energy. After that, bond formation, peptide bond formation between the first amino acid carboxyl group to second amino acid amino group with help of peptide transferase. After bond formation, what happened here? The elongation factor G, with help of elongation factor G, translocation takes place. As a result, this A plus tRNA, P plus tRNA moves left side. That means P side tRNA occupy the E side, A side tRNA occupy the P side.
Okay.
As a result, A side empty. Now immediately the same step would be continued. The same step means with help of EFTU GTP energy, another tRNA amino acids occupy the A site. Next, bond formation between second amino acid to third amino acid. Next, with help of the EFG translocation. As a result of translocation, now the A site empty. Now comes the fourth amino acid and for bond formation between third and fourth. Now translocation takes place. Now next, which amino acid comes?
Fifth.
Hello? Fifth.
Fifth.
After fifth bond formation, translocation sixth, seventh, eighth,
Yes.
until
Understood,
last.
sir. Understood.
Right,
Last nonsense codon.
right. Now reach the nonsense codon. Is it clear?
Yes, sir.
Right. Now, right. Please be silent. Please be silent. Please be silent. Keep in mute. Hello.
Okay, sir. Okay.
Thank you. Right. So this is first amino acid, second amino acid. As I said earlier, third amino acid. Third amino acid here. Next, first, third, fourth amino acid. Second. So waiting for fifth one. Right? Hello?
Sir.
Right. Now, what happened in elongation? This amino acid chain, polypeptide chain will be growing continually. But when the A site reach the nonsense codon, what are the nonsense codon? What are nonsense codon? What are the nonsense codons?
UAA, UAG, and UGA.
UAA, UAG, UGA. Right? These are nonsense codon. If anyone reach the A site, because these are not complementary to the any amino acid. So these are considered the nonsense codon. When this A site reach the nonsense codon, it will be terminated. Now let's see the termination slide. So now termination. So what will happen termination? As I said, are you follow slide?
Yes, sir.
Right. In termination, when reach the any nonsense codon here A site, immediately here also termination factors also there. The termination factors RF1, RF2, RF2, RF1, RF2, RRF. These are the termination factors. So when reach the nonsense codon, immediately RF1 bind to the A site. Later RF2 also bind to the A site. The RF1, RF2 have the GGQ, glycine, glutamine, made up of glycine, glutamine amino acids. After that, this RF1 removed, RF2 also removed, and later RRF attached at A site. As a result, this A site, sorry, E and P site tRNA become empty, removed. Now, finally added by initiation factor 3. What is the function of initiation factor 3? Already we discussed first class.
Disassociation.
Right. Now, as a
Disassociation.
result, dissociation of 30S and 50S subunits. Now, this protein strand after bind with RRF releasing factors, this protein strand, already prepared protein strand, this is called protein strand. This protein strand separated, and two subunits also separated. Two subunits also separate. Later, it forms another protein strand. The same process. It forms another protein strand. The same process. So this is about termination. Is it clear? Any doubt in this?
No, sir.
Right, in elongation process, almost same in eukaryote translation also. Okay, right?
Okay.
Right, thank you.
Thank you, sir.